First, I would like to thank the SERGG for the opportunity to present our cancer microarray data here. You just heard Dr. Vedra's wonderful talk. Along the same line, and a group of cytogeneticists, molecular geneticists, as well as molecular pathologists get together last year and formed a consortium we call a cancer. Cancer Cytogenomics Microarray Consortium. So I'm going to just give you an introduction, what we do, what we are, what we do, and show a little bit of data we have right now. So the Cancer Cytogenomics Microarray Consortium, as I said, it was established in August last year in Chicago, and our goal is to establish a platform-neutral and cancer-specific microarray design. So that design, and it's not a platform itself, but it's a content which will be specifically targeted at cancer. Right now, CCMC has its version 1 cancer array, and that's on the Agilent platform. And Illumina and Affymetrix, they are interested, but their platform is just not as flexible, and so they are still behind in their design. We also would like to share cancer microarray data between participating institutions, and we feel that is a very important educational activities. We all would like to be involved. Because of this cancer application is so new, we don't really have a lot of data to refer to. And although there are many publications, but you can hardly find any two publications give you pretty consistent result. And that's just reflection of the cancer genome, the complexity of cancer genome. And we also would like to carry out multi-center cancer genome translational research. Right now there are two major project going on. One is funded by Luminex ACMGF award this year, and that is to study the efficacy of cancer microarray application in hematological malignancies. And the other one is multi-center cross-platform clinical trial, which we pick 2 sites for each platform. Right now the sites is picked, and we're going to get into the details like material transfer, IRB between different institutions. The last but not the least is to create a public cancer array database, and which the ISCA is an excellent example for us, and we've learned a lot from ISCA. This is our website, and you can Google Cancer Microarray Consortium, and you will find the CCMC. And right now we have 166 email accounts. The last time I sent out an email through the list, and that's what they told me. And we have about 140 members, because some people may have 2 email addresses in there, and about 120 clinical laboratories. And our members mostly stationed in USA. There are a bunch from Canada, from UK, Switzerland, and we also get a lot of inquiries from Europe, other area of Europe, as well as from Asia. Now I'm going to switch the gear a little bit, well, not a little bit, a lot, and to show you why we're doing this and what's the clinical significance to try to apply the cancer microarray in cancer diagnosis. First, as what we all know, genomic aberrations are key elements in cancer diagnosis and prognosis. We all know WHO changed their classification many times since year 2000 to reflect genomic aberration and in the calculation and the prognosis in cancer. Certainly, it's very related to disease classification, and we don't really use the FAB classification for leukemia lymphoma, which was set up way back in the 80s, and that's based on the morphology. And now, and they call 821 translocation, and they call 922 leukemia, or 821 leukemia, or 5q minus leukemia, instead of M1, M2. And also, for risk stratification and treatment selection, and our clinicians will call us as soon as the specimen gets to the lab. Did you see the 5q deletion? Did you see the FLT3 mutation? So, and that is what they base to decide what treatment regimen they're going to apply to a specific patient. As we all know, there are many ways we can detect copy number variations. So without talking about cytogenetics, that's still the gold standard for cancer. However, with cancer, you can't really get 5 to 10 meg-based resolution. You probably, the best you can get 10 to 20 meg-based resolution if you're lucky. And also, definitely for cancer, for cytogenetics, you need chromosomes, and you may not always get that from cancer. And also, FISH was implemented for cancer diagnosis in the middle of 1990, and it can eliminate the requirement for metaphases, but certainly you still have the limitation how many probes you have and how many probes you can use for a specific cell, as well as this is just a targeted genome. You don't look at the whole genome. So microarray, which started to be used in clinical diagnosing constitutional disorders just a few years ago, has given us some new opportunity to get into the cancer diagnosis to help to make better evaluation of the copy number changes. So there are two different kind of cancer arrays. One's CGH-based array, the other is SNP-based array. And they each have their advantages, disadvantages, so I'm not going to go into that. But in by and large, and most of them can serve the main purpose. So this is the CCMC version 1 array, which is derived from the Tulane Haywood Genetic Center cancer array, which we developed in 2008. And this array, as what you can see here, I used an example to show you how the array was designed. And the array is a combined targeted and whole genome array. So the target here is about 423 genes here, and about 130 cancer-associated genomic regions, which we know those regions are cancer-associated, but don't really know which gene is associated, and as well as all the subtelomere regions. So it comes with 4 different designs, and I just brought in the track into the genome browser so you can see, and the 105 design, 44K design, and 60K design, and we also have 4 by 108K design. So here's the targeted region, and I used 2 genes on chromosome 11 as an example. And those other regions we targeted at exome level. So every exome will be at least have 1, and most of them have multiple probes to cover that exome. And in the case that exome does not, we can't get a good probe, and we will use the probe which is near the exome in the intron area, but it's unique. And this is the backbone region, which is the interval between those cancer-associated regions, as what you can see, and the density of that is dependent on how dense your array itself is. So about 20,000 of the genes is designated to cancer-specific probes, and the rest of them is the backbones. So the following information is a summary of our 144 cases, most run at Tulane. We now have about 200 at some cases from other labs. It's over 200 cases now. We also run 25 normal controls. The reason to run those normal controls is not used as a reference DNA, but as a separate case, is to allow us to subtract the common copy number variations, and that's present in general population. So I'm not going to go over that. And so what are the analytical criteria we use? We use relatively no stringent criteria. Algorithm ADM1, threshold 6.0. We use both fuzzy on and fuzzy off. If you use DNA analytics from Agilent software, that the purpose for that is for mosaicism. If you use fuzzy on for all of them, then you're going to, a lot of mosaicism is going to be screened out. And we look at 5 or 20 consecutive probes. And the reason we have 2 here is if it's fuzzy on, we look at 5, so we make sure we don't miss any small copy number changes. If it's fuzzy off, we look at the 20 probes, consecutive probes, so we make sure we don't miss region which is mosaically changing. We use log-to ratio between plus minus 0.1 and plus minus 0.2, that 0.2 or 0.3, and that is for the purpose of mosaicism as well. As we all know, when you're dealing with cancer genome, you are essentially dealing with mosaicism. And the software we use for analysis, in addition to DNA analytics from Agilent, we also use Nexus from BioDiscovery. Result, out of those 144 cases, we identified about 2,000 copy number alterations, and among those, and about 900 deletions, a little over 1,000 duplications. It looks like a little bit more duplications. And the size range from 2 kb to 148 megabytes, and that was a case of whole chromosome 2q duplication. And most of those copy number changes involve at least 1 gene, and 60% involves about 10 known genes, and 25 involve about 50 or more than 50 genes. I would have to say, among those over 1,900 copy number variations, although we were able to get rid of those common copy number variations, which happen to be present in normal individuals, and there are some private normal copy number variations, it's still included in that, and we haven't got a good way to get rid of those yet. I'm going to give a few examples to see what this technology can do compared to our current gold standard FISH and cytogenetics. This is a patient with AML. So cytogenetically, what we found was the patient has a 12p deletion, you can pretty much tell here, and that was confirmed by FISH. Because the reason I say it's confirmed is we normally don't run FISH panel for this probe for AML, because that is a probe for ALL, acute lymphocytic leukemia. AML is acute myeloid leukemia. But we run that, and just to be sure, because the chromosome preparation is really short, and our technologists are really, really good in our lab, and with 40 plus experience, we're able to identify 3 out of 20 cells with questionable 16p deletion. So we ran microarray, and what we found here is there is a deletion on the 12p, which is 17.5 megabytes, and it's -1, so we know that's pretty much non-mosaic. And then when we look at the 16, we do see 14.4 megabytes deletion, and it's also close to -1 log-to ratio. So we know it's not just in 3 out of 16 cells, it's probably all the cells. It's just because of the quality of the chromosome, we can't really be sure. So this is what we say, and the microarray can confirm, and what cytogenetics and molecular genetics a fish can detect, but it can also help us clarify, and sometimes we do have conflict between cyto and fish, and we want to make sure we call it the right thing. So this is a case of ALL in a young adult, and what we saw here is a small marker chromosome. So when I first looked at that, and we saw it's just an extra Philadelphia chromosome, which is relatively common and gives the patient a poor prognosis. So when we look at our fish result and we run panel for ALL, we have 922 probe, and which is a dual color, dual fusion probe. We only see 2 fusions here. That's what you can see here. There is only 2 fusions. Supposedly one is from the derivative 22 here, and the other is from derivative 9. So, and this just tell me, and this may not be the extra Philadelphia chromosome, so I call it marker. And then we ran a microarray in this case. So what we found here, and there is a duplication of the 9q terminal region destroyed to the ABL gene, and duplication of proximal region of 22, and proximal to the BCR region. And then I immediately realized, I was like, oh no, this is a Philadelphia chromosome, because there's a duplication there. And you look at the proximal region of the chromosome 9, proximal to the break point, and you see a small deletion, and an even smaller deletion on chromosome 22. So which means the reciprocal signal on the chromosome 9 is deleted. So, and that tells us, if we don't run array, then we would not know that. And also, and this just gives us, we can tell our physician this is extra Philadelphia chromosome and render the patient with poor prognosis. They probably won't treat the patient with chemotherapy. They will just go directly to bone marrow transplantation. In addition to confirm, clarify cytogenetic deficiency result, and microarray, because of its resolution, allow us to identify many small copy number changes. And this is the same patient, and as what you can see here, we saw a small deletion at T-cell alpha receptor alpha gene, microdeletion of 9q, a homozygous deletion of part of RB gene, and the homozygous deletion of part of CCND1 gene. And because we have so dense coverage in those cancer genes, we were able to show the partial deletion of the gene. Although we don't know the significance right now, there is a trend, more copy number variation in the patient with poor prognosis. However, and the immediate application in my practice is to use those copy number changes and to trace patient, to monitor the patient response to treatment, and to detect residual diseases, or to early detection of disease relapse. I have a patient with ALL, and the patient, we found by microarray there is a deletion of T-cell receptor alpha and beta gene. So the patient is cytogenetically normal, and about 50% you're going to get cytogenetically normal. So we use that marker to trace the patient. And when the physician said, oh, this patient is clinically completely in remission, but we found this deletion still present. And then we were able to detect the patient with residual disease in the same case. So we also found a lot of those small copy number changes are associated with apparently balanced translocation. In the 32 cases we analyzed with balanced translocation, 20 of them showed cryptic deletion or duplication at or near one or both of translocation breakpoint. So I'll give you an example of that. And this is a patient with AML preceded by MDS. So we found by cytogenetically, and there is a 3-way translocation and between the 2 chromosome 3s and the chromosome 12p. And then we found a marker chromosome, which is a ring, and the patient is triple X, and we did the box smear, confirm that patient is constitutionally triple X. So microarray confirmed that ring is chromosome 7. So there is a partial deletion of the P arm, partial deletion of the Q arm, and certainly confirmed the duplication of X. In the meantime, we identified 4 different small copy number deletions, and 3 of them on chromosome 3. And this deletion, which was not identified by CITO, that breakpoint, and just told us, and this is actually a 4-way translocation, involved inversion. And plot this into a genome browser that will show you what are the genes involved, and because of the density of the probe coverage in cancer gene, we were able to pinpoint where the breakpoint exactly is. So this is NCKIPSB gene, which is also called AF3 gene. It's commonly involved in the 3-11 translocation in MDS or AML. And this is a gene, 3Q-RPN1 gene, and the EBI1 gene. Those are involved in the inversion of 3Q, commonly seen in AML or MDS in myeloid disorders. So, having said all these good things about microarray, but still, in my view, at least for the time being, microarray is an adjunct test, not as inconstitutional. We can't just based on microarray for interpretation. To be able to give a comprehensive characterization of complicated genome, we need probably all of them. So this is a patient also with AML preceded by MDS. As you can see, there is a 5q deletion, and there is a 16-12 translocation unbalanced, and there is missing a chromosome 19, and derivative 20q, as what you can see there, there is a big unknown material add there and a marker chromosome. I want to emphasize that chromosome 19, it's missing 1 copy. So this is the report table I sent to our physician. The copy number, the microarray confirmed 5q deletion, but it doesn't look like all the 5q is deleted, and it's not consecutive deletion, and part of the 5q is not deleted, and also, and there's the amplification or duplication of part of the 5q. Same for chromosome 12. And chromosome 12, as what you can see there, there's 2 deletions, not consecutive, and also there are 2 duplications. I forgot to tell you on the other slide, and on the chromosome 12p, there is a gene called TEL, and that gene is supposed to be deleted. Instead of deleted, FISH showed amplification. So, and that is where the gene located, P13.2. That's the TEL gene located, and that's because there is an amplification in that region, and that's why the FISH showed normal or amplified result in some of the cells. And as you can see, and there is no deletion of chromosome 19. So chromosome 19 must be somewhere in the genome. So we went to back to look at figure out it with FISH. So these are the deletions on chromosome 5. This is the duplication on chromosome 12p, and that's the chromosome 16q deletion, and all those are confirmed by FISH. So what you can see here is we use chromosome painting. Chromosome 5 is green, and chromosome 12 is red. As what you can see, part of the chromosome 5, this is the 5q deletion, and part of the 5q is translocated to the 20q, the chromosome 20q, and the other part is incorporated as part of the marker chromosome. And the same work was done for chromosome 19 and 16. And we see this is normal 19, and the other part of the other chromosome 19 is translocated to the 20q as well as the marker chromosome. Okay, so I'm almost there. So just go through this. And this slide, following a couple slides that just showed the capability of this array system to detect mosaicism. This is cases we know by FISH mosaicism. And as what you can see, if we know it, we probably can tell 7%. But if you don't know it, let's see this. And we did the dilution theory. And this is for duplication. If it's below 20, and it will be hard to detect. We probably can, for deletion, to detect 15%. And this is for deletion. And this slide shows the inter-experimental variation. And as you can see, and these are different lot number of array, and you can see the pattern is almost identical. So there are limitations of CGH arrays, and the major limitation is copy neutral, loss of heterozygosity. And we have some of our cases sent for SNP array. We ran this at Dr. Wolf's lab with the Illumina array. And this is a patient with MDS. And 3 years ago, we found this patient with -7. And now it has been for the last 3 years chromosomally normal. And when we ran the SNP array, we found loss of heterozygosity on almost the whole 7q, and which is the critical region for chromosome 7q. So there are other limitations. Oh, by the way, Agilent is coming out with SNP probes, and so we hope we can incorporate that into our design for the next version. Balance translocation, and so far there's no platform can detect that yet. We probably will have to wait for next year or some other technology come out. Mosaicism. And there are limitations to detect mosaicism, but maybe for solid tumor, we can do tumor cell enrichment. For hematological disorder, right now, we pretty much use for new patient and at relapse. Constitutional copy number variations, one reference DNA we can use patient reference, patient normal cell as reference. It may not be possible for hematological disorder unless you use skin biopsy. So the Cancer Cytogenomics Microarray Consortium is going to hold a second meeting in August on the 5th and in Chicago again. We have invited UCSC Genome Browser, DBGAP and DBVAR to give us presentation, and hopefully we will follow the discussed steps and to help to set up a database. So this is an acknowledgement. Most of this work is done by Dr. Xiaofeng Hu at Hewan Genetic Center. Terry is sorry. Any questions?
So, can I ask the first question?
Yes.
So, in the variations of unknown significance, where you see an abnormality in an abnormal child and you don't see an abnormality, you see the abnormality in a normal parent, how do we know, and is there a role for sequencing the allele, the other allele? Because there may be, of course, a point mutation that's not picked up by array that could render the patient with an autosomal recessive condition. And
that's certainly a possibility, but it's not yet cheap to do that, and it's not yet a clinical fee-for-service test to do that. So on a research basis, there are a lot of people thinking about that, but it's, I think the best answer is most of the time, if you can really get your hands on that, quote, normal parent, they're not normal. So it's still pretty rare.
Which of us really are? But I guess
No, no.
I guess the point is the ones that we put into the category of benign variants may subsequently need to have that other allele at the deletion site sequenced before we can really say that.
So the ones that are in the normal databases now as benign, certainly if you have that, if it's a deletion, there's always the chance of a point mutation causing a recessive disease. So they're not 100% benign in terms of reproductive risk for somewhat increased reproductive risk for an autosomal recessive genetic disease. But it should show an autosomal recessive disease phenotype when we get to the point that we know all of the autosomal recessive genes and clinical phenotypes.
David, I just have one sort of quick question as far as recommendation or prediction maybe. Given how quickly everything is evolving right now and trying to move these variants from one side to the other, how do you envision things playing out really in the next, say, 3 to 5 years as far as all these patients that have had arrays and have findings, and how are the labs going to go back to all the information and either take the array that was run and say that the platform's not going to change, so you got to go back and maybe continuously revisit the information or the interpretation and start trying to make more than you can make from it right now?
Yeah, so that's a tough question because most labs don't have good database retrieval systems, and their written reports may not include all copy number changes that were interpreted as most likely benign, and so they weren't followed up, etc. So going back to your patients when something becomes known to be pathogenic is difficult for most clinical labs. I think when we start submitting data to these central databases and all the raw data files, then it's easy for researchers, but also for clinical labs to reanalyze their own data. So it will be a tractable problem in the future. And the arrays, I think, although they're still evolving pretty quickly, I mean, we're starting to cover a higher percentage of genes, and the backbone resolution is already at a point it's more than we need. So I think we'll start slowing down the rapid change in the platform, and you'll have a lot of data that you could go back and reanalyze or reinterpret.
Do you envision having something sort of been decided upon in the database, being able to say, okay, these are all these cases that match this particular finding, and having the people that submitted those cases actually be notified?
I was asking whether the database will actually have a functionality that when something goes from uncertain to pathogenic or benign, you can actually send a message back out to the submitting labs to alert them that they should reclassify. I haven't thought of it that explicitly, but that would be a nice advantage to the submitting labs to have that sort of feedback. So it's a good idea.
I think it's critically important that these consortia continue, and I'm glad that they're put together, and I'm also glad that you're keeping an eye out for the future with the whole genome sequencing and looking at the model for this as a way to help with that. Because excited as I am about that, I also have an overwhelming sense of dread about what those reports are going to look like and how we're going to use them in the clinical setting. But something that Marilyn alluded to, and I was just curious, is the issue of mosaicism. And I'm just wondering how, in the constitutional situations, has that been evaluated, or has it been a problem in assessing, the importance of CMVs and the thresholds based on mosaic presentation?
Most of the platform thresholds are in this 20 to 25% mosaicism should be detected. It depends a little bit on the size of the deletion and the duplication, but in general, 20 to 25%. So you're not going to get really low 5 to 10% mosaic. So if you had a clinical suspicion of something in particular, that might be a case where cellular fish would allow you to look at hundreds of cells, whereas it may be below the threshold for an array.
So I think that will be the last question.
This question, is there the NCBI database for the phenotype, is it just static, just one-off, or are you planning to have a longitudinal phenotype dump into the database? Like, for example, how many of those may go ahead and develop a malignancy cancer some type in future? Or is there a overlap between the two?
Yeah, I mean, everybody's ideal is to update phenotypic information if you see the patient back and have more information. That's going to depend on the volunteer efforts of submitting times and or grant funding to support updating phenotypic information going forward. So it's hard to know exactly how successful that will be.
Thanks very much to both of those speakers.